If you would, open your books to chapter 7.
Jordan, can I get your lab book later?
7.3 and 4 really is what we're doing today. We already did 7.3, but we're going to review it and then go to 7.4. So what page is 7.3 on? 24. I just want to do a quick review of what we talked about yesterday, or Friday, I guess it was.
Oh, it's not anyone.
A quick review of the light reactions of photosynthesis. And for some reason, this is some of the hardest stuff. So you should be focused during this class period here. Hold on, let me get these magnets down.
Did you make all of these magnets?
Thank you. I like to play with my magnets.
What?
All right. Let's keep in mind what we're trying to do with photosynthesis. What's the object of photosynthesis?
To create food and plant.
Not to create energy.
Make glucose.
To make food, glucose. It's to trap the energy from the sun in a molecule, glucose. And this is what glucose looks like. There she is. And so we're going to end up making this after we're all done. Do you all know the formula for photosynthesis?
6 CO2 plus 6 H2O is
solar
energy.
Sunlight.
Solar
H12O6 plus 6O2.
H12O6.
Okay, you got to know this. You need to know this. Now, the book put 12O2 on the left. I'm sorry, 12 H2O on the left and 6 H2O on the right. But if you knock 6 H2O off both sides, you end up with this equation. And then, yes, you need some light. Or you can put light over here, I guess. Light energy. And so this is photosynthesis. Carbon dioxide and water make food and oxygen. That's a quick way to say it. Carbon dioxide and water make food and oxygen. Where does the water come from?
The roots.
The roots. When you water your plants. Where does the carbon dioxide come from?
Air.
Floating around. Okay? So, this is basically photosynthesis is how that happens. Now, we want to see, where do you think, any idea where this is happening in a plant? Someone except it, besides Isaac?
Cycloid.
Where are the chloroplasts?
Cycloid.
Where are the thylakoids?
And the granum.
Where are the granum?
Stroma.
In the chloroplast. Where are the chloroplasts? In the plant cell. Where are the plant cells? In the plants. In the leaves of the plants. So it's happening in the leaves. Okay? It's happening in the leaves. And, well, that's the wrong one.
Oh, what else is
We want this.
Computers can do chlorophyll. More like chlorophyll.
What movie?
Billy Madison.
Okay, so we're looking at a leaf. And when we look down into a leaf, we see all these leaf cells. We call them mesophyll cells. Those are the ones with chloroplasts. And if we look down at a chloroplast, this is what a chloroplast looks like. It has these membranous sacs called thylakoids. And if we're looking up close at these things, there's a thylakoid membrane. And that is this. That's where we are right now. Do y'all see where we are? Savannah, you with me? What do we call the area outside the thylakoid membrane?
Stroma.
The stroma. Okay. And all of this, remember, is in a chloroplast. So, what happens? Well, this is review from last time, so you should already know this. So if you don't, pay attention now and make sure you ask questions. But what happens is light shines down, hits the chlorophyll, and that energizes electrons. They kind of jump out of the chlorophyll here in photosystem 2 and start moving down this electron transport chain. And as the electrons move down the transport chain, hydrogen ions, also called protons, get pumped inward by active transport. They get pulled in this way. And so every time that happens, every time electrons go down, hydrogen ions get pulled in. We're going to build up a large concentration of hydrogen ions down here, you'll see. Are you with me? Kind of sideways. The electrons that left the chlorophyll have to be replaced, and they're replaced by water molecules that are nearby. So here's a nearby water molecule. The electrons go into the chlorophyll, and that water molecule breaks apart. Then the electrons become energized and start moving down the chain. More hydrogen gets pumped in. And that's going to continue to happen.
How does hydrogen get pumped in again?
There is a protein right here that uses the energy from the electrons to pull in hydrogens.
Where do they come from? The water?
The hydrogens? Yeah, they're originally from water.
So like once they're done jumping, like I don't understand where they are, how they're outside.
Well, and what happens is after we build up all these hydrogens here on the inside, they're going to go out.
And they get pulled back in.
And then they can come around and get pulled back in. Nice. Other questions? Yes.
Now, all those H pluses, does that make it acidic?
Very acidic.
I see.
Acidic in here. That's how they discovered this is happening. They did a pH test on the inside of the thylakoid. And that's some of the evidence that this is going on. Other questions? So, every time a water molecule breaks up, the hydrogens are released, and now you end up with just oxygen. And the two oxygens will stick together, and that makes O2. That's the oxygen that's produced in photosynthesis.
Wait, so the H plus can like just go out and then like come right back in?
Yes.
Just like keep being
These are just protons, and they once they get pumped out, they can move around and they can come back in.
It can just like forever be reused.
Yes.
Like it never stops working or anything?
It would stop. These things would stop moving around if you quit getting sunlight. If it was dark, then they would kind of equalize and just kind of bounce around and wait till there was more sunlight.
What happens at night? They just chill?
They're just chilling. What happens is we have a concentration difference right here. And so after these get pumped out through the ATP synthase complex and you get an even distribution on both sides, then you have no net difference. So it's kind of like an isotonic situation. you have the same amount of hydrogens on both sides, so it's just kind of remains like that until the sunlight shines again. And then electrons will start coming down, and then these things will get pulled back in. You understand there's a whole stack of these things, right? There's one, and there's another one under it, and another one under it, and another one under it. So this thing can go out here and go in the next one down. Cool. Something else can happen to the hydrogens. At the end of the electron transport chain, after the electrons are re-energized again right here, the electrons end up getting picked up by NADP. NADP is a molecule that we call an electron carrier, and it picks up these electrons, and it'll pick up a hydrogen too. So now the hydrogens are picked up, and we now have a new molecule called NADPH. So some of the hydrogens can end up there.
What does that do?
That's what we're going to talk about next.
What is an ADP?
It is a molecule that is high in energy. It has high energy electrons and hydrogens, and that will be used to make glucose in the Calvin cycle. Nicotinamide adenine diphosphate. You don't have to memorize that. So, Caitlin, did you see where some of the hydrogens can end up? Picked up by the NADP. It's like a little taxi. And it comes and it grabs these electrons, boop, and grabs a hydrogen, boop. So hydrogens are getting picked up from the outside, and hydrogens are getting pulled into the inside. And whenever water breaks up, it leaves some hydrogens on the inside. So you end up with bunches of hydrogens on the inside and not very many on the outside. And they will go through this ATP synthase protein. And every time a hydrogen goes through, that powers the coupling of an ADP and a P to make ATP. So hydrogen will go through, ATP will be made. Hydrogen will go through, ATP will be made.
Are there just a ton of ADP and Ps hanging out?
Ton of
over there?
ADP and Ps just floating around.
What are the yellow squares?
Electrons.
These are electrons. Those came from the water molecules. Right?
Can you zoom in on this?
Yes.
How?
The up arrow. That's it.
What does the ADP take from the hydrogen that makes it ATP?
Nothing. It's just energy. As this comes moving through here, it kind of somehow causes the ADP and P that had previously bound to this protein. As the hydrogen comes through, there's energy released to make ATP. It's kind of, I've heard it described as like the turning of a water wheel. As the water goes by, it turns the wheel, and this thing kind of turns and puts the ADP and P together somehow.
So it's ATP synthase.
ATP synthase means to synthesize.
So it's the way the protein is shaped that lets it.
Yeah, it has something to do with the physical shape of it.
What are the things that let hydrogen in?
They're little proteins here in the membrane. You don't have to know the names of those proteins. Proteins in the membrane pump in hydrogen. They have names, but they're not important. Yeah? We hadn't gotten to that yet. That's in the Calvin cycle. We're about to do that.
And how does water go?
Water is just pulled into the plant by the roots. And it goes everywhere in the plant cell.
So it can just go. It doesn't have to go anywhere. It just goes in.
Yeah, it's just pulled into the plant cell. When we study in a later chapter how that works. But as long as you keep water in your plants, water will go into the plant cells. But they do have to have water. That's important. You can see, without water, none of this would happen. Are we good? You want to see a little video footage of it? I mean, an essay, part of the essay for the next test might be describe the light reaction of photosynthesis. Bam.
Okay, one more question.
You got to write all that.
When the oxygen stuff together, where do they go?
They just float out. They
So
just
they
diffuse out.
can just go through
Those
the
oxygen-
Oxygens can go right through the membrane.
Don't they go through the stomata?
Yeah, so they can't get out of the leaf unless the little stomate's open. Have y'all met stoming?
No.
Y'all haven't met stoming?
Stoma's like Stoma's those little holes on the bottom.
Oh, in the leaf, right?
Hi, I'm stoming. I'm a little hole in a leaf that opens to let in carbon dioxide and let out oxygen.
What's it called?
Stomate. It's my little puppet here. Stoming. I talked about stoming in the first video, I think, right?
No.
I didn't even talk about
You
it?
haven't talked about them yet.
It's lame. I'll talk more about stoming later. Ooh, let's just see a little video footage of the light reactions here.
In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. Now
hold on, they kind of went through that. See that right there? That's the ATP synthase that I have down here on the bottom. But it could easily be right here next to all these. And they have it spinning. So as the hydrogens come through, it spins around, and that puts together ATP somehow. A cell biologist would know how. I don't.
Are those shooting stars sunlight? Yeah.
The shooting stars are the light.
Which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH.
So they're just kind of showing the whole thing going on here.
Is it photosystem 2 is the force photosystem 1?
Photosystem 2 is the first one in the chain.
So, when the hydrogen hooks on the 2 electrons in the NADP,
Yeah?
it's NADPH.
That's right. And NADP becomes NADPH when it picks up the hydrogen.
And when it already has the electrons, right?
No, the electrons come from the chain.
Yeah.
Yeah, it kind of picks them all. It kind of docks here and sticks here to this protein. And the electrons will attach, and then a hydrogen will attach, and when all those attach, it kind of changes its shape a little and undocks.
But before it's called NADP.
It's called NADP originally,
Okay.
and then NADPH when it picks up the hydrogen and the electrons. Okay? So let's go to the Calvin cycle then and make some glucose. We've done the light reactions. This part's called the light reactions, and it made NADPH and ATP. Now we're simply going to use that NADPH and ATP to make glucose, because these are high energy molecules. They can get work done. And so the next part, I've got all these videos. This part right here, which is in your book, you can see it's a cycle. Whoops. You refer to this picture in the book, page 128. This is called the Calvin cycle. The guy who discovered it was named Calvin. And it basically happens in 3 parts. And there's a bunch of different names of chemicals, of molecules here that you could memorize, but you don't need to. You won't be tested on it. Even on the AP exam, they're going to ask you the names of these things. But here's basically what happens. Sitting out here in the stroma are molecules, and one that there's actually 2 molecules that you should know out of all of these. And one of them's called RUBP. That stands for ribulose biphosphate, or ribulose bisphosphate.
What is it?
RUBP.
But what is it?
It's a molecule that sits out here in the stroma, and it consists of 5 carbons and a bunch of hydrogens and a bunch of oxygens and a bunch of phosphates, a couple of phosphates, and other things connected to this. But I'm just going to show the carbons in my example because I don't want you to get too confused. So here we have a 5-carbon molecule. What's it called?
RUBP.
Nice. Is that EKF?
What do you know about? You know the name of it?
I'm gonna teach it. Hold on. I'm gonna teach you about what it does. RUBP is sitting out here in the stroma. And CO2, carbon dioxide from the air, is going to diffuse in when those little holes in the leaves open. Sucking in air. There are little holes in the leaf that can open up and let carbon dioxide in. And here's the carbon dioxide. And it's going to float in here and combine one molecule at a time to the RUBP. Now, see I have the letter C? This stands for a carbon dioxide molecule. These things floating around here, these are CO2s. I'm just showing you the carbon. You have to imagine there's also oxygens connected to it. But I don't feel like putting all those in there because that would get confusing. It'd be too much stuff on the board. It's already enough stuff on the board. So can I just leave that there and you pretend it's carbon dioxide? So the carbon dioxide comes in and attaches to the RUBP molecules. And that's what this first step is. It's called CO2 fixation. See the carbon dioxide coming in and attaching to the RUBPs? And they come together to make 3 C6 molecules. Now it's not just C6, it's like C6H12O3 P2. I mean, it's a bigger molecule, but we're just worried about the carbons right now.
Wait, can you say
Well, I got the first step and then you.
Here we go. Three CO2 molecules come in and attach themselves to 3 RUBP molecules, forming 3 6-carbon molecules. When I say 6 carbon, I mean they have 6 carbons per molecule. 1, 2, 3, 4, 5, 6. That's this process we call CO2 fixation. The CO2 fixes itself onto the RUBP. Becoming fixed is becoming attached.
3 6 molecule what? What are they called?
See these 3 things?
Yeah.
Count the number of carbons they have.
And it's 6. So it's 3 6 molecule RUBP molecules?
It's not RUBP anymore
What is it?
after this attaches.
6 molecule.
It's a 6-carbon molecule.
Thank you.
All right? Then these 3 6-carbon molecules, they're kind of unstable and they kind of fall apart a little bit. Boop, like that. And now we have 6 3-carbon molecules. 6C3. And they have a name. They're called phosphoglycerate, but you don't need to know that. There they show 3PG, they're calling it. So, going back over that, the CO2 attaches itself, and then the molecule kind of falls apart. Now we have these 6 3-carbon molecules.
Okay, split in half.
Yep, split in half. Very good. These molecules will be attacked by the ATP and the NADPH that we made earlier in the process. And hydrogens will be given to these molecules, and electrons will be given to these molecules, and energy will be given to these molecules from the ATP. And the ATPs fall apart back into ADP and P. And so these molecules, these 3-carbon molecules that we have get changed, you see. By ATP and NADPH, they get changed into a new 3-carbon molecule. And that molecule is called G3P. And that's the other molecule you have to know for this cycle. Just these 2.
We don't have to know the BPG.
You don't have to know either of these, no. So G3P is glyceraldehyde 3-phosphate. And that's what we have right here. These are now called G3Ps. We got 6 of them.
What's the name of it?
G3P.
What does it stand for?
Glyceraldehyde 3-phosphate. You see it's on this table right here.
We don't have to do it.
Oh.
What's different about it than before?
They now have energy that the ATPs gave them. They have electrons and hydrogens that the NADPHs gave them. We created NADPH and ATP in the light reactions. We have now used them up to make these molecules higher energy and contain different things.
To do what?
We're going to make glucose eventually. We're on our way to making glucose. It's exciting, isn't it?
What's that step called?
Glucose.
No. What is that step called? The first one was
Carbon.
CO2 fixation.
We call this CO2 reduction, this whole step. Reduction means you're getting electrons added to you, and it reduces your charge. Did y'all learn about oxidation and reduction in chemistry? Were y'all awake at all last year?
I don't think we did.
We didn't get taught that.
Yeah, y'all did that.
We all failed.
We got taught what?
Oxidation reduction reactions?
No.
I don't think. I really don't think we learned that.
You just had
You did that, you just don't remember. Anyway, we call this CO2 reduction when the electrons the electrons from NADPH are given to these molecules. Now we have G3P. Moving on, can we move on?
Yeah.
Yes.
One of the 6 G3Ps is stored away. See right here? One of the 6 G3Ps is stored away, and now we only have 5 G3Ps left.
Where is it stored?
Just right here. It's just going to sit right there for just a minute. Can y'all just wait a minute and just let it sit right there, and just enjoy the fact that it's right there. And now we have 5 of these G3Ps, which is not as many as what we started with, what we just had. And these 5 G3Ps are going to be rearranged by enzymes and more ATP input from the light reactions. So more ATP comes over here. I dropped my P. There it is. More ATP comes over here that was just made here in the light reactions and supplies more energy, and that energy is used by enzymes to rearrange these 5 things.
Wait, why are there only 5 now?
Because we stored one away, and you're to enjoy that. Just sitting there. So ATP comes in, and some enzymes use the energy from ATP to rearrange this. I want you to watch me rearrange it. It's exciting. You ready for the rearrangement?
Ready.
Pew,
No.
pew, pew.
1, 2, 3. So now there's 3, 5.
We're back to the 3 RUBPs we started with. See?
What happened to the little one over there?
But don't they? Oh, wait.
Just enjoy it.
It's a little.
Just enjoy this. Don't try and force anything onto this yet.
Okay.
Oh,
Yes.
oh, stretching.
Okay, I want to do the whole thing again. I want to do the whole thing again. That's what I want to do. You ready? Here we go. Here are the light reactions. The Calvin cycle. 3 CO2s float in. They attach to your RUBPs. Now we have these 6 carbon molecules that break down into 3 carbon molecules. NADPH and ATP change them around a bit by giving them energy and electrons. One of the 6, they're now called G3P molecules. One of the 6 is put away. Just enjoy it there. And the other 5 are changed back around by more ATP coming in, changes it back around into the original RUBPs we started with.
The 3, 5 carbon molecules, which are RUBP.
So once it builds up enough of those...
Ah!
Oh,
Stop!
sorry.
You're not enjoying. I want you to see what we have down here after we've done that twice.
Can you?
Can I what?
Can you wait. Okay.
Let me do it again.
The 3 goes down. Okay, I get it.
You got it? Okay.
Can I ask a question?
Yes.
Okay. Well, for like one of my first steps, it says I wrote oh, never mind. Got it.
Okay, here we go. We have 2 G3Ps just stored down here, right?
Yes.
Am I right? If y'all obviously weren't enjoying them if you don't remember that.
Yes.
They're going to be hooked together. And now we have a hexagonal 6-carbon molecule, glucose. Bam, we make glucose.
so it takes 2 run-throughs of the cycle to make one
Where'd the
glucose
second one
molecule?
come from?
Bam!
Hexagonal
Okay, sounds good.
A hexagonal 6-carbon molecule, which you already know is...
Glucose.
Glucose.
We just made glucose. Took us 2 days of instruction. Yes.
Where's the second?
one of the still recover.
Okay, each time CO2 attaches, then they break apart, right? Then NADP and ATP, NADPH and ATP change them around, and then one gets stored away. And then ATP rearranges them to what you start with. And then the whole thing will happen again. 3 carbons come in, break apart, they're rearranged, and another one gets stored away. So now you have 2 that are stored away next to one another, and they will connect to form glucose. There, I've got 2 glucoses now.
So that's C6H12.
This molecule is C6H12O6 because it's not just carbons here. There's other stuff attached to them that I was telling you have to imagine.
So is it a different type of glucose, or is it just there's just glucose?
Just glucose. Now this glucose can be changed to almost any organic molecule. You put a whole bunch of glucoses together, you know what you have? Cellulose, starch. If you unwind the glucose and straighten it out, you got a fatty acid tail. If you hook a couple extra things to it, you have a nucleotide, and so forth. This is the basic organic molecule that can make everything else. And that's the Calvin cycle.
I feel like that's way more complicated than
I mean, it's how it is.
I know.
How would you do it?
I don't know. I wouldn't do it.
I wouldn't.
How would you make glucose from H2O and O2?
No. Like I'm just saying. Like I feel like it like takes apart, it comes back together, and it takes apart, and it goes together. Like so many times.
It doesn't really take anything apart. We're just putting together. I guess these are put together then come back apart. These NADPHs, you're right. But they're just taxi cabs, you see. The NADPHs will go back here after they've dropped off, and they'll pick up more electrons and more hydrogens, and then they'll just bring it over here and drop them off. So they're just grabbing things and dropping them off.
Okay, can I ask a question again?
Yes.
So if you want to tell us.
Oh, wait, one more thing. Read 7.4. 7.5 you don't have to do. 7.5 is out.